Assalamualaikum and good day to everyone. Okay, so today we are in the last session for the revision and discussion for the answer scheme knowledge booster. And for the last chapter, okay, we are going to discuss about the coordination. Okay, so coordination is considered the second major topic for your PSPM because it consists of about 15 marks. Okay, and the last chapter in our syllabus, okay, 11, which is the immunity. So marks allocate for your final PSPM is about 6. All right. Okay, so let's start our discussion. Okay, so this is the question for coordination. Okay, if you recall back, okay, coordination. Right, so for the first question, okay, so the figure 1 shows the formation and transmission of an impulse along the axon of the neuron. Okay, so if you recall back, okay, when we are discussing this transmission of the impulse, actually we are discussing about the resting potential, action potential, the presence of the sodium voltage gated channel, potassium voltage gated channel. Okay, so whenever you get the question, so please identify what is the related subtopic that you can trigger your idea before start answer the question. Okay, so let's identify the potential that we have here. Okay, so the first one will be the, change the color here. Okay, so this one will be the resting potential. Okay, so this is the resting potential. So which is uh, the charge for the resting potential will be the negative 70 millivolt. Okay, all right, then this one will be your threshold level. Okay, so if you still remember, threshold level where the trigger of the stimulus will occur here. Okay, so if the stimulus able to reach until negative 50 or 55, so they will be undergo the next process here will be, the next stage will be the depolarization. Okay, all right, so I push here, depolarization, and the maximum of the depolarization will be the action potential. Okay, all right, so the maximum the charge for the action potential will be plus 40. Okay, so plus 40. So the next stage here will be a repolarization. All right, and the last one will be the hyperpolarization. So please make sure you understand what happened in each stage here. All right, so let's look at the question here. So the first one, name the phase P, Q, and event R. Okay, so P will be resting potential. Okay, the second one, Q, which is the threshold level, or also can write as a threshold potential, and R. Okay, so it's a action potential. All right, done. Okay, so now let's go to the next one. Okay, so you have to explain what is happened in each stage that you mentioned above. Okay, so for example, like in P. Okay, so as you know, P is a resting potential. So when it's a resting potential, so you can start your idea with P is resting potential. Okay, so during resting potential, what is happened? Okay, means no impulse is transmitted. Okay, why no impulse is transmitted? Because more negatively charged inside the axon compared to outside. Okay. All right, then why? Why is more negatively inside the axon compared to outside? So due to both, okay, both sodium and potassium, the voltage gated channel is close. Okay, all right. So actually, if like this, 6 marks is given, so you can estimate. Okay, so each question they will be give you about 2 marks. All right. So now let's go for the second one. Okay, which is the threshold level. So during threshold level, actually, what is happened? Okay, so the stimulus, okay, is triggered. Okay, so okay, causing some, okay, only some, not all, okay, some of the sodium voltage cathode channel to open. Okay, so what you can understand, when sodium voltage cathode channel is open, so they will allowing some of the sodium ion to enter inside the axon. Okay, oops, dah lebih dah. Okay, sodium inside the axon. Okay, so what happened? So bila some of the sodium enter inside the axon, so now will reach the charge up to negative 50, negative 50 or 55. Okay, or negative 55. Okay, so it's a range lah. All right, due to the presence of the sodium inside. Okay, so now let's go for the R. Okay, so as you know R, okay, maksudnya the maximum of the depolarization. Depolarization, okay. So what happened here? So you can understand here. More, okay, so you have to give the comparison from the threshold level. So more sodium more sodium voltage cathode channel is open. Okay, so more voltage sodium voltage cathode channel is open, so allowing more sodium to enter inside the axon. Okay, so what happened now? More positive charge is produced inside the axon compared to outside. Okay, so bila there is more positive charge inside the axon compared to outside, so impulse can be transmitted. Alright, so done. Okay, so you just have to recall back all the information when we are discussing about each stage. Okay, so like that. Alright, so what will happen if the depolarization is less than negative 15? So what happened? If less, means the stimulus is not strong enough to open the some sodium cathode channel, so they're not allowing a lot of Na to enter. So the charge will be less. So they will be back to resting potential. Okay, so they like a merajuk lah. Alright, so no impulse will be transmitted. No impulse will be transmitted. Alright, so done. Okay, so it's just like a this one is referred to the all or none event. Okay, so all or none. Okay, so let's continue. Second question. Okay, for the second question, if you look at here, Okay, so it's a transmission of the impulse in the neuromuscular junction. Okay, so if you understand or you just recall back, neuro represent the end of the or synapse here. Okay, end of the neuron. Okay, muscular here represent the muscle. Okay, and why it's in the form of junction? Because it's like a T junction there. Okay, that's why neuromuscular junction. Okay, so here this one is the neurotransmitter. So you must write here as a acetylcholine, acetylcholine. Okay, which is your neurotransmitter inside the neuromuscular junction. So neurotransmitter. Okay, so inside the inside the muscle lah. Okay, so X is what? Alright, so you can understand this one is the impulse lah. Okay, so if you want more specific, alright, so this one will be the motor end plate. Okay, where the sodium voltage gated channel all is located here. Okay, which is the beginning of the muscle. This one is the sarcolemma. Okay, C will be the T tubule, where your impulse will be moving down across the T tubule, and here is the sarcoplasmic reticulum. Okay, so sarcoplasmic reticulum, and here this one will the sarcoplasmic reticulum is consists of calcium. Okay, ions. All right. So then the calcium ion will be enter. So this location is known as a sarcomere. Alright. Inside the sarcomere consists of two filament. Which one? This one is known as an actin or thin filament, and this one is the myosin or thick filament. So see, it's very easy to understand if you just look at the diagram and you're able to identify all the structure here. Okay, so we already identified what is your A. Alright, so this is the answer lah. Okay, so acetylcholine or neurotransmitter. Alright. Saya naikkan ke atas. Alright. So what will be your B? Okay, B mana B? See, this one is the calcium ion. Okay, so if let's say you want to write in the formula, so make sure calcium 2 plus. Alright, so then the next one, what is the C? Okay, C is the T tubule. So ingat je, nampak dia macam T shape. So it's a T tubule. Okay, make sure huruf besar T tubule. Alright, what is the types of a neuron found in this neuromuscular junction? Okay, so if you recall back when we are discussing about this coordination, okay, even though we have a 3 neuron, but we are focused on motor neuron. Okay, so that's the answer. Your answer will be the motor neuron. Okay, name the two types of myofilament. Okay, can you recall back what mean by myofilament here? Yes, whatever that is located in our sarcomere filament, which is undergo the sliding filament theory. So we have a actin and also myosin. Alright, so these two myosin. Okay, so actin and myosin. Alright, done. So what is the role of B in the calcium contraction? Okay, what is your B? See, your calcium ion. So what the calcium can do? Okay, so calcium will bind to troponin. Okay, so troponin dekat mana? So look at here. So here, so if you look at here, this one is the actin. Yang small bits ni is actin. This one tropomyosin, and here this one is the troponin. Jadi calcium will bind to the troponin at the actin or thin filament. Okay, so what they will do, exposing the myosin binding site. Okay, So bila they exposing the myosin binding site, so what happened? Alright, so the myosin which is come from the thick filament, they will be able to bind at the actin. Okay, so they will start to the contraction. So other than this one, so actually we have another function of the calcium. So another function of calcium to stimulate the ATPase. Okay, so to hydrolyze the ATP into the ADP and phosphate group. So this one will cause our myosin head with the high energy configuration. Okay, so that's why they can uh, forming as a cross bridge. Can you still remember the cross bridge? Okay, so myosin head, this is the actin, and they can form or attach to the actin. Okay, so that one is another function. Okay, so here calcium will be bind to the troponin and exposing the myosin binding site. All right, and you can add that one also lah for the calcium. Okay, so describe the event occur at X. Okay, here. So as you know, here the impulse is transmit, okay, or moving. And what is your Y? Y? Alright, so it's about the sarcoplasmic. Okay, so let's discuss the X. So what is happened? So here, there is actually action potential is propagated, okay, along the plasma membrane. Okay, membrane. Okay. Okay. So other name for your plasma membrane is in what? It's a sarcolema. Okay, sarcolema or sarcoplasmic. Okay. So never mind, I just put as a plasma membrane and enter the tubule. Dah stop. Okay. So the next one. What happened inside the T-tubule? Okay, so if you look at here, consists of the calcium. So what is the location? Which is the sarcoplasmic reticulum. So action potential. Okay, triggers the sarcoplasmic reticulum. Okay, sarcoplasmic reticulum to release the calcium to plus. Alright, so to release the calcium to plus. Okay, done. Settled for the one question. Okay, now let's go for the third question. Alright, so this one is one of the question yang considered quite tough for the student because you have to memorize the light band, the dark band. Okay, so this is the repetition for the myofibril. Okay, so this one is showing the one side of the sarcomere. Okay, so now name the repetition. Okay, so I mentioned just now. So this one is the sarcomere. Okay, sarcomere is a block of sarcomere. Okay, state the theory used. Okay, so if you recall back, so it's like a sliding filament theory. Okay, sliding filament theory. All right, then. Okay, so describe what happened during muscle is contraction. So if you look at here, this one is the thin muscle. This one is the thin filament and this one is the thick filament. So what happened? A band. If you look at here, this is the A band, di mana the location of the myosin or thick filament. So as you know, thick filament is constant. Dia maksudnya dia won't move. Dia tak akan bergerak langsung. Dia static di situ. So what happened? Then they will be remain same. Okay, so they will be remain same. Okay, done. Alright, now next, what happened to H zone? Look at here. Where is your H zone? Okay, so it's actually H zone. Mana dia H ni kat sini? Ni dia H zone. Okay, so H zone maksudnya is showing between the gap 2 of the thin filament ni. So kita nampak bila muscle is contract, so the both thin filament they will be sliding to each other, tapi they won't langgar they just sliding like that. So what happened? The the H zone will become narrow. Okay, so dia mula narrow lah. Dah mula sempit dah. Okay. I band, nampak? I band also same because kita tahu the thin filament is moving. So, they also will become narrow. Okay. Narrow. Okay, so, okay, look at here. So now identify first. So this one is the myosin head. Okay. Alright, and this one will be the troponin. Okay, so as you know, inside the troponin contain with the calcium. Okay, calcium binding site. So where your calcium will be bind here. Okay, binding site. Alright, so calcium. Okay, and this one is what? Okay, so kita nampak it's like a dot. It's like this. So calcium lah ni. Okay, so kenal pasti dulu calcium. Okay, so nampak it's like a small bits like this. This one is the actin. Okay, alright. Then this one is what? Okay, tropomyosin. Tropomyosin. Okay, so done. Actually, kita dah siap identify whatever is a request in our question. So A is a actin. Okay, so you just write back actin. Okay, then second, what is your B? Tropomyosin. Okay, and what is your C? Troponin. So kat sini jawab troponin, tropinin pula sorry, troponin. Okay, so it's a troponin. Okay, it's a troponin. Alright, so explain the function of D. Okay, so what is the function of D during the muscle contraction? So the D, okay, so just write as a D lah, will bind to troponin and causing the tropomyosin to change the conformation. Okay, so bila dia dah change the conformation ataupun shape lah. Okay, so dia macam akan terbuka macam tu Okay, so dia akan what they do, exposing the myosin binding site. Okay, then. Okay. Alright, so that's all. Okay, so let's go for the last question. Okay, so this one is represent the hormone on its target cell. Okay, so if you look at here, this is the hormone and this one is the plasma membrane here. So if you look at here, our hormone is not simply diffused into the cell or target cell. So if like this, so clearly this one is the non-steroid hormone. Why this one is a non-steroid hormone? As you know, non-steroid is non-lipid soluble. Okay, so means they cannot simply diffuse inside the plasma membrane. So they have to use the another pathway. Okay, so kita dah nampak. Alright, then let's go to the question. Okay, what mean by hormone? Okay, it's one of the chemical messenger. Okay, which is secreted. Okay, by cell or gland. Okay, so we have a pituitary gland and so on. Okay, which will be transported into bloodstream. Okay, bloodstream and function at target cell. So chemical messenger dihasilkan dekat gland lain, masuk dalam darah dan berfungsi dalam target cell. So you ingat dia punya flow like this. So senang untuk kita faham. Alright, so one characteristics of the hormone which is enable it to enter the target cell. Okay, this one refer to our figure 4. Okay, so what is the characteristic dia? Dia adalah water soluble. Okay, or this one is the non-lipid soluble. Sama lah. Okay, so you can write as a water soluble or non-lipid soluble. So what types of hormone is shown here? Okay, so saya dah tulis what types of hormone. Kita hanya ada dua, which is steroid or non-steroid. So this one is an water soluble, so this one is non-steroid. Okay, tu je. Right. So can you recall back what is the example for the non-steroid? So it might be insulin or glucagon. Okay. So ingatlah beberapa contoh. Okay, name the structure A. Okay, so kalau kita nampak, this is the hormone. So A will be the hormone receptor, which is dia akan stay dekat our plasma membrane. So hormone receptor. So once hormone receptor bind with the hormone, they will be formed hormone receptor complex. Okay, so what will be the B? Mana B? Okay, enzyme. Enzyme apa? Only one enzyme is adenylene and what? Adenylene cyclase. Adenylene cyclase. Okay. And what is the C? Okay, so dia akan adenylene cyclase will trigger the catalyze the ATP. Okay, to become CAMP ataupun second messenger C, pastikan C kecil ya, AMP. Why this one is a second messenger? Sebab your hormone is the first messenger. Okay, that's why yang ni adalah second messenger. Okay, messenger CAMP. Alright, nampak. Okay, now briefly explain. So actually masa kita bincang ni, Madam dah sebut sedikit. Okay, so now kita jadikan dia ayat yang formal lah. Okay, the hormone. Okay, so actually sebenarnya kita tengok je balik dekat sini. Apa hormon ni buat? Okay, dia bind. Alright, so the hormone bind with hormone receptor, to form hormone receptor complex. Okay. To form hormone receptor complex. Alright. So the binding will activate siapa? Nampak? Siapa yang duduk kat sebelah ni? Selepas dia bind dia, kita nampak tak? Nasib baik dia dah labelkan untuk kita. So they will activate the G protein. Okay. The G protein. Okay. Titik. Alright. Now, what the G protein will do? So G protein pula will activate adenylene cyclase. Okay, cyclase. Okay, so what the adenylene cyclase will do? Sebab dia adalah enzyme, will catalyze. Okay. The ATP into CAMP. Okay. So now C, walaupun dia mula tapi tetap C tu huruf kecil. The CAMP act as the second messenger. Okay, dah. Alright. So banyak markah. The hormone bind with the hormone receptor complex, satu markah. The binding will activate the G protein, second mark. They will activate the adenylene cyclase, third mark. They will catalyze the ATP into CAMP, fourth mark. The CAMP act as a second messenger. Nampak? So all together will be the 5 marks. Okay, so now let's go for the last chapter in our biology semester 2, which is the immunity. Okay, so if you look at here, alright, so before we identify, so look at the structure here. Okay, so if you look at here, it's a L shape. So ingat, kalau L shape is a light chain. Senang sebenarnya kalau kita nampak. So this one you nampak is like a H. So this one will be H represent the heavy chain. Heavy chain. Okay, so Z. Okay, so this is the area or the location. Besarkan sikit ke sini. Alright, I move here. Nampak? So this is the location where alright. So this one is the area where the antigen binding sites. Okay, so antigen binding site. Alright, so nampak SS. Okay, so this one is the bond which is a disulfide bridge. Okay, disulfide. Disulfide bridge. Okay, now name the molecule. So this one is the antibody. Okay, ataupun kalau secara scientifically known as immunoglobulin. Okay, or short form is the IG. Sorry, I besar IG. Not Instagram. Okay, identify the structure. That. Okay, so this one light chain. Okay. Alright, so X, where is your X? So this one is a disulfide bridge. Y, nampak? So dia nampak macam H, so this is the heavy chain. And Z, this is the antigen binding site. So bila antigen binding site ni yang nanti antigen dengan dia mesti akan comparekan based on epitope. Epitope, one antigen can be represented by different the active site, okay, for the antibody to bind. Okay. Alright. So define the antigen. Alright. So as you know, antigen is what? Okay, a foreign. Okay, a foreign which trigger the immune response. Okay, so kalau dia tak berjaya trigger the immune response, so it's not the antigen. Okay. Alright. Two interactions. So actually altogether we have three. So kita namakan three, right? Neutralization. Okay, so we have opsonization, and we have activation of complement system, and pore formation. Alright. Okay. So what is the inaccurate to refer the pathogen? Okay. Maksudnya, kenapa kita sebut antigen to antigen? Sebenarnya kita tahu antigen is come from the pathogen itself. So why we never represent pathogen? Kenapa? But we are always mentioned as an antigen. Okay? Sebab bila the pathogen is a whole structure. Okay? So because the pathogen because the antigen is a part only. It's a part of pathogen. So it's not the whole structure of the pathogen is involved here. Okay. So it's only represent a part. Okay, a part only. So dia bukan keseluruhan structure. That's why kita tak boleh panggil dia the whole pathogen attached to the antibody. Tak boleh. Okay, so it's a small part. Alright, and normally bila dia adalah antigen, it's normally mostly large protein molecule. Okay, so that's why dia tak boleh disebut sebagai pathogen. Sebab pathogen sometimes can be a virus or bacteria. Okay, so let's go for the final question. Alright, so if you look at here, showing the P and Q represent the level. Okay, so here primary and booster dose. Okay, antibody during the primary and secondary immune response resulting from the vaccination. Okay, so what is the relationship between antibody and antigen? So antigens, antigens are molecule, okay, that trigger the antibody. Okay, antigen yang trigger the antibody. Kalau tak antibody ni buat tak tahu. Okay. Okay, so which one is the primary? So kita nampak which one will be the primary. So X. And which one will be the secondary? Y. Sebab kita obviously nampak bila ada booster dose. So definitely this one is the secondary. State the class of antibody represent. Okay. So kalau dia adalah primary. Okay. So ingat IG, mama. Okay. Mama always come first. Okay. Lepas mama datang, barulah girlfriend datang. So IG. Ingat, apa-apa, mak datang dulu. Mama dan IG will be the secondary. Okay. Explain the differences between primary and secondary immune response. So, we can talk about the time, is it long lasting ataupun the rate. Okay, so let's say I start with the rate. A rate of response is in primary immune response are slower, okay, compared to secondary. Okay, secondary. This is the first differences. Then the second one, the effect in primary response, in primary immune response is short life, while in secondary immune response is long lasting. Okay. All right. Why the person usually not infected by the same pathogen more than twice? Okay. Kenapa dia takkan? Because the memory. Okay. So maybe memory T helper. Okay. T helper will activate immediately. Okay. Immediately to attack the pathogen. Alright. Okay. Alright. And give one example of the disease commonly vaccinated against. Macam semua vaksin yang kita pernah ambil lah. So can be TB, okay, can be rubella, and nowadays can be COVID. Okay. So that one all is the vaccine which is can represent as the this one. Okay, for the secondary. Okay, so that's all. Okay, so we already cover all the questions under knowledge booster, okay, in the SAL book. All right. So I hope whatever that we are discussing and all the answer scheme that we discuss will help you to score better, and the main thing is actually you able to identify the question and able to answer perfectly your final PSPM. So here I would like to wish all of you good luck. Okay, so may God bless all of you and easy all your final paper, especially for biology PSPM 2 SB025. So Good luck, everyone. Okay, thank you. Assalamualaikum. Bye.